all. I'll see if I can share my screen. Brilliant. The components of Zoom bingo. I don't know if you guys know that game. Another one is, can everybody hear me? So can everybody hear me?
Yes.
In my lab, we study genetics of psychiatric illness. And I'm going to give a brief rundown of some of the work that's come out of our lab in about the last year and what it is that's different about doing this kind of work now than it was even a couple of years ago, and the huge advances that the field has made and why it's such an exciting place to be now. So just very broadly, the goals of my research group are to identify risk loci for psychiatric and substance use traits, mostly using GWAS or genome-wide association studies. We use deeply phenotyped local samples that we've collected here over the past decades. We have about 20,000 such samples here, but we also use very large national and international samples. And we have a paper under review now with a sample size over 1 million, which is kind of cool. But we don't just want to identify the largest number of risk loci. Of course, we want to do that because that's great to have more risk loci than anyone else. But the point is to use that to understand disease biology, mostly via post-GWAS statistical analyses and epigenetics and other techniques. And then ultimately, the goal is clinical translation, which we see signs of being able to do in the near-ish future by means of pharmacogenomics and polygenic risk scores. Now, I'm going to use some terms like genome-wide association study that I will assume people understand. But if you don't understand these terms yet and you're interested in the work, just contact me after the talk and I'd be happy to talk with you. So this graph is from a slide from Pat Sullivan that came out a few years ago, showing the relationship between number of risk loci identified and size of a genome-wide association study. And what it shows is a fairly consistent pattern of, first there's what's called a dead zone, where you get a few thousand or even a few tens of thousands of subjects, and you get none or maybe 1 or 2 risk loci. But then you pass a certain threshold, which differs by disease. And once you pass that threshold, you can expect a consistent pattern of discovery, where every time you add N cases, you add Y risk loci. So we're in that region now for traits like schizophrenia, where every time 1,000 affected subjects are added to GWAS, we find about 4 new risk loci, which is pretty exciting. A decade ago, there were only a couple of risk loci for schizophrenia. The traits that I'm going to discuss are earlier on in this curve, but are past what we consider the inflection point, where we're consistently identifying more and more risk loci. The huge progress in the field in recent years has come from massive meta-analysis and biobank samples. So one of the well-known biobank samples is the UK biobank, which has 500,000 subjects collected in the United Kingdom, relatively high SES sample. But the data are made very widely available, and therefore there's been a huge amount of discovery from it. And they have self-report surveys and electronic health record, or EHR linkage. One of the problems for psychiatric traits is that this is a well-educated, high socioeconomic status group, and therefore they don't have a whole lot of substance use disorders and schizophrenia, and the kind of traits that are of greatest interest to us. But it's still a magnificently useful sample. Another example is 23andMe, which I'm sure you've all heard of. It's a direct-to-consumer testing company. They now have millions of subjects, and we're working on a project for a trait I can't mention with some other investigators, where there are close to 3 million 23andMe subjects included. And then there's the Psychiatric Genomics Consortium, which is one of the largest biological collaborations in the history of the world, and that's a meta-analysis group. So they have been responsible for a lot of the forward progress in psychiatric genetics and genomics over the last decade. They are the origin of the great Schizophrenia 108 Risk Locus paper, which came out in Nature about 5 years ago. One of their most interesting papers in the last year was from their anorexia work group. And one of the outcomes of that paper was that it showed that anorexia is as closely allied to endocrine and metabolic disorders as it is to psychiatric disorders. So that resulted in a very interesting reframing of the understanding of the disorder. One of the large samples that my group works with most is the Million Veteran Program, which at present is less well known, although that's starting to change. So in my group, we have co-lead of several MVP projects, including the Post-Traumatic Stress Disorder Project. We have the data from the UK Biobank. We have collaborations with 23andMe, and I'm one of the leaders of the Psychiatric Genomics Consortium Substance Use Disorder Work Group. So we have a lot of cool data. So what can we do with that cool data? So a bit of the information about some of the available data sets and what we can do with them. This is a description of the Million Veteran Program project, which you can find on the MVP website. The web address is given on the top of this slide. So it's a national voluntary research program funded by the Department of Veterans Affairs. And the idea is to increase our understanding of the genetics and genomics of disorders that are prevalent in military populations. So diseases like diabetes and cancer, but also military-related illnesses like post-traumatic stress disorder. And if you're a post-traumatic stress disorder researcher like me, then you might be really excited about gaining access to the MVP sample, because all of these people are military or ex-military, and they have a lot of traumatic exposures like combat exposures, and there's a lot of information in that sample. So far, 825,000 subjects have been recruited. We have genetic data from over 300,000 of those already. And generally every year we get a new tranche of about 150,000 subjects. We expect to get more data next week. And the MVP, being a government project, has a very high inertia. And the consequence in this case is they decided when they reach 1 million subjects, they're not going to stop. They're just going to keep recruiting at least to 2 million. So this is a fantastically useful sample. And if you want to read about it, the article header that I just showed is the basic paper reporting on the structure of the MVP and what it's for. There are a lot of great characteristics of the MVP. It's got lower socioeconomic status than several of the other great biobanks. I'm not going to talk about most of the advantages, but I'll just point out there's representation from non-Europeans, which addresses a major problem in our research field, where most of the data are from Europeans. So there are a lot of African ancestry and Latinx subjects in MVP, and that's proved to be enormously useful. I'm going to show you a few examples of data from our group in the last year. So first, I'm going to discuss genetics of alcohol use disorder, which is a chronic relapsing brain disease. The heritability is about 50%. For a couple of decades, there have been a few well-known risk loci that map to alcohol metabolizing enzymes. And this is the first major work product of the PGC-SUD Substance Use Disorder Working Group. It was a trans-ancestral GWAS of alcohol dependence. It came out in Nature Neuroscience, December 2018. And so as of December 2018, there was only one alcohol use disorder risk locus in European ancestry subjects, another risk locus mapped to the same gene in African ancestry subjects. So keep this in mind. It was only a little less than 2 years ago, only one risk locus. And this was actually a cover story of Nature Neuroscience. I really love this figure. It's called plot glasses. It's chromosomes 2, 3, and 4 from our GWAS. My daughter was the graphic artist, by the way. So on to the first Million Vets program study of alcohol use disorder. In this study, we considered two phenotypes. One of them was alcohol use disorder per se, which is a dependence diagnosis, a measure of physiological dependence, which indicates tolerance and withdrawal symptoms. And the other was a quantity frequency measure, which is called the audit C. So that's just how much a person drinks. And I'm not going to talk in a lot of detail about that, but the important difference is alcohol use disorder, heavy drinking, medical consequences, and physiological dependence. Alcohol quantity frequency, the audit C, indexes a trait that occurs more in the normal range. So prior to our work with these phenotypes, and this is the paper that came out in Nature Communications last year, prior to this work, many people thought that alcohol use disorder and quantity frequency measures of alcohol use indexed the same phenotype or a very comparable phenotype. But we found was that, first of all, we raised the number of alcohol use disorder risk loci from 1 to 10. And second of all, we found that of those, only 5 overlap with quantity frequency measure. And this slide is my favorite from the paper. The analyst was Hong Zhao, who's associate research scientist in my research group. And this compares genetic correlations with other traits by linkage disequilibrium score regression, by audit C on the top and alcohol use disorder on the bottom. And the pattern of correlations is very different. So in other words, audit C has one set of genetic similarities with other traits, and alcohol use disorder a very different set. So audit C is showing negative genetic correlation with anthropometric traits like body mass index, and negative correlation with coronary artery disease, which is that it seems to be protective, whereas it shows only negative or no correlation with psychiatric traits. Alcohol use disorder shows positive correlation with all of the major psychiatric traits we looked at. So this is an example of where having all of these genetic risk loci on a genome-wide level is now enabling us to dig much more deeply into the biology of these traits than we've ever done before. I want to leave a little time for questions, so I'm just going to rush through the rest of my slides. We had a paper in Nature Neuroscience in June, which included many more samples for alcohol use disorder. Hang Zhao did the analysis for this too. We had a very good pickup in the media from this paper. And now we're able to go much more deeply into the genetic correlations with other traits by means of doing the PheWAS with the Vanderbilt University BioView data set. And again, I'm not going to linger on this. If you're interested, give me a call or send me an email. And then finally, by meta-analyzing our local and our MVP samples, we've now gotten a robust genomic hit for opioid use disorder. And this came out in JAMA Psychiatry also in June. And for the first time ever, by using MVP data, we're now able to look at the genetic correlations of opioid use disorder with other psychiatric and medical traits. And that's what this slide shows. Finally, we also did a study of PTSD re-experiencing. And I'm just going to show one thing from that. And this shows the utility of looking at non-European populations. The top slide is European ancestry. This is our GWAS of PTSD re-experiencing. There is a long linkage disequilibrium shelf, which corresponds to a chromosomal inversion that's common in European populations. In African ancestry subjects, we have much lower statistical significance. It's a smaller sample, but this localizes. And when we meta-analyze the European and African ancestry subjects, we now have a localized signal with 2 orders of magnitude localizing support, which maps to CRHR1, corticotropin releasing hormone receptor 1, which in years past was a major candidate locus for post-traumatic stress disorder. And we're only able to get the localizing information because we have data from both European and African ancestry, because we can't localize in European ancestry because of the inversion. The inversion is much rarer in African ancestry. When we meta-analyze them, we now have localizing information. So that's just one reason that it's great to have non-European population information. And this paper came out in Nature Neuroscience also last year. So we have a lot of ongoing projects. Some of them are psychiatric genomics consortium work. We're working besides with the SUD work group, with the PTSD work group, the depression work group, the ADHD work group, and we've given data to several others. We have a lot of work going with the MVP. We have 2 decades worth of research in Thailand, where we have a long-term training program, and now we have a methamphetamine genetics program, and we have a specific program in alcohol use disorder in African ancestry populations in the US. And that's just some of the work that we're doing. Here are some of the major collaborators for the work. And I'll leave some of the projects on the screen, and I think we probably have 2 or 3 minutes for questions if anyone wants to ask any.